Hello everyone, myself Dr. Vivek Kumar, Professor, Himalayan School of Biosciences, Swami Rama Himalayan University, Jollygrant, Dehradun. So I am with you to discuss few aspects of endophytes. So my broad topic is endophytes or endophytic microorganisms. In this topic, I will discuss with you about endophytes, how they make an entry inside the host plant, how to isolate the endophytes from the host plant, how to identify them, and then the effect of endophytes on the host plant. So in this picture we can see that there is a plant, and there is above ground plant as well as the below ground part of this plant in the form of root. And when we enhance or increase the size of the root, we can see there are certain microorganisms which are present outside the root, and some of the microorganisms which are present inside the root. And another above picture shows that even the microorganisms, they are present near the root hair, inside the cortex. So they are present inside the plant, are known as the endophytes. So we can say these are the organisms. They could be bacteria, fungi or actinomycetes. So they live between the living plant cells. So the term endophyte is derived from a Greek word endon. Endon means inside or within, and the phyton means plant, we know that. So within the plant or something inside the plant is known as the endophytes. So earlier the endophytes, they were defined as the microbes such as bacteria and fungi that inhabit the plant endosphere. Endosphere means the inside environment of the plant during all or part of their life cycle without causing any apparent harm to the host plant. It means if the endophytes are present inside the plant, then they are not causing, if they cause, that is not apparent, they are not causing any harm. So definition of, we can say, this endophytes has been changed from time to time. So recently the endophytes, they are defined as microbes including bacteria, archaea, fungi, protists that colonize the plant interior regardless of the outcome of the association. Normally we believe that once the microorganisms, they are associated with the plant, then they should benefit the plant, they should augment the growth of plant. But sometimes the endophytes, they are present inside the plant, but they are, they are not that active, metabolically active. They are present, they are live, but they are not giving any observable effect on the host plant. Again, this picture shows that there is a plant, and when we increase the size of the root, so we can see certain microorganisms outside the root, in the soil it means, and some of the microorganisms, they are present inside the root plant. These are known as the endophytes. So how to isolate them? So conventionally the endophytes, they were isolated from surface sterilized plant tissues and cultivated in a nutrient rich medium. It means that whenever we want to isolate any type of endophyte, then from any part of the plant, it could be root, it could be stem, it could be any twig, it could be seed, it could be flower or leaf. So first we have to surface sterilize the sample material. The surface sterilization will remove all the epiphytes, the microorganisms which are present on the surface of that sample. We are interested in microorganisms which are present inside the sample or inside the tissue. So in recent years, many endophytes have been identified through culture independent approaches. That is the metagenomic approach. So such as sequencing of 16S ribosomal RNA gene, the internal transcribed special region ITS1, ITS2, through whole genome sequencing of endophytic community. So there are many host plants which have been reported to have or to possess different types of endophytes. So these are the examples of the bacteria, for example Azochus, Burkholderia, Gluconobacter, Klebsiella, Pentoia, Herbaspilum, etc., etc. So these type of microorganisms, they are commonly present inside the plant. This picture shows that we can say that these are the fluorescent pictures. This picture shows that in the first A, it is in the red part of the plant part, yellow dots are there. So the yellow dots, they are the fluorescently tagged endophytes. And here the green portion, dark portion in the other pictures, we can see that these are the endophytes which are present inside the host plant. Now, there is a process of recruitment of bacterial endophytes by the host plant. So, before that, let's understand what the rhizosphere is. The rhizosphere, if you define as the soil root interface where the complex interactions take place between the plant and surrounding soil microorganisms. So, it has been reported that plant release significant amount of photosynthetes, this is also known as the root exudates. And these photosynthetes, they influence microbial communities in the rhizosphere. The rhizosphere means, for example, this is the root hair. The immediate contact area of the root hair is known as rhizoplane. And then slightly away from the rhizoplane is the rhizosphere. And the rhizosphere zone is always in the soil. So, the immediate part after the rhizoplane, when the root exudates are secreted by the roots, that soil region is enriched with photosynthetes. And that's photosynthetes, it contains lot of nutrients in the form of organic acids, minerals, amino acids, proteins, carbohydrates, etc., etc. Because of this rich region, rhizosphere region contains lot of microorganisms. Even we can say the diversity of microorganisms. The root exudates likely contain substances that initiate early communication between the host plants and the bacterial endophytes. And consequently, this process steers the colonization process. Colonization process means the bacteria which have been the rhizobacteria, once it's inside, it makes an entry inside the host plant, then it becomes the endobacteria. For example, evidence of involvement of oxalate, this chemical, in the recruitment of the bacterial strain Burkhardia phytoferments PSGN by host plants have been reported. So, it means that oxalate play a role in the recruitment of the Burkhardia species. So, in a study conducted by a scientist, this phytoferments strain defective in oxalate utilization. It means the bacteria was not able to utilize oxalate produced by the plant, was used to inoculate lupine and maize plants that secrete moderate and low levels of oxalate. Then what happens? Let's see. The mutant was observed in significantly less numbers in both maize and lupine after 3 days of inoculation as compared to the wild type strain. It means the Burkhardia strain was not able to utilize oxalate, and we have discussed that oxalate is very important in making a communication or entry inside the plant. So, the bacteria was not able to make utilize oxalate, so it was not able to make an entry, or we can not able to inoculate the plant root. On the other hand, the wild type strain of Burkhardia, which was not a mutant, it was able to utilize oxalate. It was able to inoculate the plant. So, inoculation of both wild type and mutant strain result in significant differences in colonization by the two strains in lupine but not in maize. So, again lupine mein hua, it was not happened in maize because of the difference in the host physiology. So, oxalate was also observed in Brachypodium root exudates, and high number of proteobacteria were detected in the Brachypodium rhizosphere. Moreover, there is a process known as bacterial quorum sensing. So, bacterial quorum sensing compounds are likely involved in communication with the plant root and the subsequent colonization process. Again, so the colonization process of the bacteria and the host plant, here the process quorum sensing also takes place. So, importance of these plants, or we can say compounds, in the colonization and growth promotion of plants by endophytes is supported by a recent study, which is this. In this study, it has been reported that a quorum sensing mutant, Burkhardia phytoferments PSGN, could no longer efficiently colonize Arabidopsis species and did not promote its growth. It means quorum sensing is very important in making a contact between the host plant and the bacteria. So, the bacteria was Burkhardia, which was a mutant of quorum sensing, it was not able to colonize the host plant. So, when it was not able to colonize the host plant, it means it was not able to enhance the host plant growth. Now, is there any role of host plant in recruitment of the endophytic microorganisms? Yes. So, plants are likely directly involved in quorum sensing. So, some plant extracts have been shown to have quorum quenching also. So quorum sensing and quenching are different things. So quorum quenching is utilized to prevent the entry of the pathogens. So quorum quenching capabilities which could protect them against phytopathogens, and some quorum sensing molecules have been shown to have direct plant growth promoting effects. So native soil composition and the host plant genotypes are also considered important in recruitment of bacterial endophytes by the host plant. So type of soil, number one. Number two, the type of the host genotype. So these are the two main parameters. Other parameters are also there, we will discuss later. So these are the two parameters which are responsible for the recruitment of bacterial endophytes inside the host plant. So there is a study of root endophytes of Arabidopsis grown in different soil. It has been reported that soil type likely influence the composition of bacterial endophyte community found in the host roots. So this indicates that different soil types may be inhabited by variable bacterial populations that serve as initial inocula. We can imagine, for example, this is the field, we are growing for Arabidopsis or X plant here and here, and obviously when soil is different, the soil is different and the nutrient composition is different, that nutrient composition will also influence the soil microflora. So obviously when plant will be grown there, we will find the different microflora in the different locations of the same plant. So the study also showed that an environmental conditions such as soil nutrition, soil moisture, soil temperature and the host genotype and the age of the plant, they have a direct influence on root and leaf bacterial communications. So the nutrition we understand, moisture we understand, temperature also. Temperature means if the temperature is there, the temperature will decide the type of the microorganisms, mesophilic, psychrophilic, thermophilic microorganisms. So diverse bacterial communities were reported in grassy species like Dactylis, Festuca, Lolium under different management regimes. Fertilizer application, the moving frequencies indicating that agronomic operations may influence bacterial endophyte recruitment in a cultivated plants. Now, the endophytes that live within the plant are beneficial to their further development. Now this picture shows, the left picture shows that there is a no inoculation of endophytes, microorganisms. The right hand shows that the plant has been associated with the endophytes, and when these microbes are there, they are promoting better growth to the plant. Why? Because they have primed the plant. They are preventing the plants from biotic stress, they are preventing the plant from the abiotic stress, and when they also provide nutrients to the plant as compared to uninoculated plant. So obviously when the plants are getting better nutrients, plants are prevented from the biotic stress, plants are prevented from the abiotic stress, then obviously the host plant is going to show better growth as compared to the uninoculated plant. Now, the process starts with the attachment of bacterial endophytes to the host plant surface. So, attachment or adhesion of bacterial cells to the plant surface is considered the first step of the colonization process. So, bacteria in the vicinity of plant roots most likely swim towards the root, but if they want to swim towards the root, obviously here the moisture becomes one factor. If there is no moisture, obviously it will be difficult for the bacteria to move toward the root. So once they move to the root using chemotactic affinities for root exudates, it means the roots, they have secreted some chemicals, and these chemicals, they have attracted the microorganisms, it could be bacterial fungi, from the rhizosphere zone to the inside the plant. So this is followed by the attachment to the root surface. So once they move toward the root, which is getting access to the potential entry sites at lateral root emergence areas or opening caused by wounds or mechanical injuries. So normally it has happened that the plant roots, they are very intact. So there are no pores, there is no hole which allows any foreign material in the form of bacterial fungi to make an entry inside the root. But during the formation of lateral roots, or some wound has been caused, or some due to mechanical injuries, so some opening becomes, through these openings the microbes, they make their entry inside the plant root. And then exopolysaccharides synthesized by bacterial cells may facilitate the attachment of bacterial cells onto the root surface, may be important in the early stages of endophytic colonization. It means the exopolysaccharides of microbes, they play a role in colonization. So EPS produced by endophytic bacterium Gluconoacibacter was reported as an essential factor for rice root surface attachment colonization. So colonization history of rice plants using Gluconobacter showed that bacterial cells were shielded from oxidative damage by exopolysaccharides. So the oxidative damage present there may be secreted by the roots. So these bacterial cells, they are protected by this exopolysaccharide secretion. So we can say that EPS becomes crucial for the colonization process. Additionally, free radicals concentrations in planta were decreased by the application of EPS. Colonization was reduced in EPS knockout. So any strain that is not able to produce EPS, then it has been observed that they were not able to make proper colonization. Interestingly, this reduction in colonization was rescued by addition of EPS produced by another wild type strain. So bacterial structures such as flagella, fimbriae, cell surface polysaccharides, they are also involved in attachment of bacteria to the plant surface. When we study the colonization of maize plants in a report, so Endophyte herbaceolarum observed that bacterial lipopolysaccharides is necessary for attachment and subsequent making an entry, and then bacteria becomes endophyte. Later it was also demonstrated that binding of N-acetylglucosamine of LPS with maize root lectins is required for bacterial attachment and subsequent colonization inside the plant roots. Finally, the key points of this our topic is that endophytes, they are the organisms which live between living plant cells and do not cause any disease. Endophytes are ubiquitously present in diverse plant species. So it has been shown that they have been co-evolved with the host plant, and different classes of microspecies like bacteria, fungi and actinomycetes, they also colonize the internal plant tissues, and when they colonize, they lead to diverse implications on the host plant. And one can imagine that the medicinal plant associated with the endophytes have been extensively studied as a potential source of bioactive compounds produced by these endophytes. And these bioactive compounds, they have pharmacological attributes and they may have huge biotechnological applications. Now, about other features of endophytes, we will discuss in another lecture. Thank you so much.